Okay, got that going, and then I'll start letting everyone in. Hi, everyone. We're gonna give it, I want to say 2 more minutes, and then we'll get started. Dr. Connie Cepko is already here. It's a huge pleasure to be welcoming her. And I hope you guys are looking forward to the talk. Hope you guys have questions. Hope you guys did the reading. Yeah, we'll give it, 2 minutes.
Chapter books that you read.
Okay, cool. I think people will continue trickling in. We're still missing a few people, but let's get started first in the interest of time. Dr. Cepko, I'm gonna intro you super quick, and then I'm just gonna hand it off to you.
Sure.
And for everyone, the structure for today, Dr. Cepko is just gonna talk a little bit about her journey into science, and then she's gonna talk about her research, and then we'll open it up to Q&A afterwards. That sounds good with everyone? Okay, cool. Dr. Connie Cepko is the Bullard Professor of Genetics and Neuroscience at Harvard Medical School and Howard Hughes Medical Institute investigator. And this basic intro of Dr. Cepko's work definitely won't be doing it justice, but in short, her lab studies the eye, in particular how it develops very early in the first trimester in humans, the basic processes that allow the eye to develop, and what goes wrong such that a person is either born blind, or has the risk to develop blindness later in life. And the lab also studies and attempts to develop therapeutics to prevent the onset of blindness. And yeah, am I missing anything in that intro, Dr. Cepko?
Good. That's good.
Perfect. Okay. All right. Then I'll hand it off to you, and we'll get started.
Okay. Well, hello, everybody. It's really nice to see so much interest coming from high school kids. Really appreciate that, and I hope you keep up your interest throughout your lives, because it's a absolutely wonderful career to be able to ask questions about nature, how things work, and even at some point maybe address some of the underlying disease problems and come up with therapies. So I have an absolutely wonderful career path. Encourage anyone who's interested to keep on. And in terms of how I got started, I had a bit of an unusual early path. So when I was in middle school, we had to do science fair projects. This was in Maryland. And at the time, I wasn't interested in science, but everybody had to do a science fair project. So I asked my biology teacher for suggestions, and she handed me this magazine, kind of a science for kids magazine. And one of the experiments in there that I thought was interesting was growing yeast. You probably know yeast is something you can use to make bread, make beer, etc. but growing yeast in the kitchen was possible. So I did an experiment growing yeast on different kinds of carbon sources, different kinds of sugars. I don't even know how I came up with this idea of different sugars, but I did. And then in our science fair, we had judges that would come around and judge our projects. And so my judge really liked my project and asked me if I wanted to visit his lab. And since I was in Maryland, there were a lot of government labs around. This was near Washington, D.C. And so I visited his lab, which was this idyllic setting. It was called the USDA Forest Disease Lab. And his group were all about identifying tree fungi. And the lab was like a little white clapboard building in a forest. And so it was this beautiful setting. And I still remember walking in and seeing a really lovely old-timey kind of glass display case with tree fungi in it. I thought that was very cool. And then he actually taught me how to do microbiology. So he asked me, are you interested in learning more? And I was. And amazingly enough, he invited me to come out on Saturdays, and he would spend Saturday morning teaching me how to do things in the lab. So that was my introduction, and I did that for years, and I absolutely loved it. And I credit John Palmer, the man who taught me these things, with my ability to do science. I think the earlier you can do science, the better. So after that, I went to University of Maryland for my undergraduate. My family, by the way, had no money at all. So I paid for everything. By going to a state university, it was great because the fees weren't very high, so I didn't have to take out loans. And I worked all the summers and school vacations to earn money to pay for things. And so that was really worthwhile. And at the University of Maryland, I majored in biochemistry and microbiology. And I got interested in viruses. And from there, I went to MIT to graduate school. And I should say that I know that people have a lot of pressure about what college they go to. My daughters did, all their peer group did. I think it's way, way too much for people who are in high school to worry about that. I think as long as you go to a decent college, you can go anywhere from there. Just work hard, get a good education. If you're interested in science, work in a lab. But it doesn't, when I went to University of Maryland, it was considered a mediocre school. Now it's much better, but at that time it was considered pretty mediocre. But I went again because I could afford it. So if you're worried about which college you should get into, just worry about getting into a decent one. Most state universities are absolutely fine, but other schools too that don't cost a lot, as long as you work hard and learn. And again, if you're in science, work in a lab, get good letters of recommendation, get good grades, you can go to a top grad school. So that's what I did. So I went to MIT and I worked in Phil Sharp's lab working on viruses. And then I stayed at MIT and became a postdoc, and I worked again on viruses. I made some of the first retrovirus vectors, which are being used for gene therapy now. And from there, I went to Harvard to the Department of Genetics. And as you can hear, then my background was all about viruses, but I wanted to switch. And the reason I did is that my field of gene therapy with viruses was getting crowded, and I didn't want to feel redundant. And I thought about, well, why do I like biology? And I thought, well, there's a lot of great, big unanswered questions.
Tablets. So if you're sitting in the X row,
I think you need to mute whoever that is. Anyways, I thought, what are the big unanswered questions in biology? And I didn't know anything about the brain, but I thought the brain has all kinds of big unanswered questions. How do we think? How do we form memories? How do we form emotions? All of that. And so I started sitting in on a class at Harvard Medical School. So I was at MIT, but Harvard Medical School was very gracious and said I could sit in on their classes. So I did, got interested in the brain and the nervous system. And then I was lucky enough to be hired at Harvard Medical School in the Department of Genetics, which was a brand new department. And I've been there ever since. And we started studying the brain, and different parts of the brain are accessible for studies of development. And I was interested in development. And so the eye is actually part of the brain. Most people don't realize that, but it's actually part of the brain. So we started with the eye, we're still on the eye, and we studied mostly its development, and we studied other parts of the brain too, just not as intensely. And while we were studying the development of the eye, we realized that some of the genes we were studying also led to blindness if they were a mutant in people. And so we thought, well, let's look at what happens when those genes are mutated And is there anything we can do to prevent people from going blind? So now we do both things. We still study how does the eye develop, and then we also try to figure out why do people go blind, and can we address that? And so my talk will consider those questions. But I wanted to also give you a broader view of gene therapy. So at the beginning of the talk, I'll give you a little bit more fundamentals about how we're using gene therapy as a field, and a few of the applications, not just to the eye, but also to the blood. There's some really exciting developments there, and there's some also for cancer, but I didn't want to cover too much in the short time I have. So I'll just give you some examples of some success stories, how people are going about this. And please ask me questions. I understand you can raise your hand, which I'm very happy to be interrupted at any time. And also at the end, there'll be lots of time for questions. So it's up to you when you want to ask questions. So before I start, any questions from anybody about anything? Okay. So I need to have a permission from you for screen sharing?
Yeah, let me do that. How's that looking?
Yes, I can do that. Okay, does that come across okay?
That looks great.
Okay, so again, I'm going to cover a little bit more broadly the topic of gene therapy for the first part. And I'll say right at the outset that it's got fantastic possibilities. There are also serious challenges about some of the things we do, and there's great success stories. So I'll try to cover all of that. And this initial picture is about photoreceptors, which I'll tell you more about. But I'll start with this video. This is the first approved gene therapy in the United States. It's for a disease called Leber's congenital amaurosis 2. So when people are born with a disease gene that leads to this, they can't make a small molecule called 11-cis-retinal, which is important for vision. So this boy, Corey Haas, is from Western Mass, and this is before his gene therapy for that disease. You can see he's going in. He has a cane. He goes to a school for blind kids. He can only read with Braille. So he's affected pretty severely, and he has been from birth. Now, after his treatment, so he has one eye injected with a virus, and I'm going to tell you more about the viruses, but this virus delivers the gene that's defective in Corey. And so here's how it looks after he gets treated. Okay, so you can see that that worked really well. And this is inspirational to my lab. So this was first approved in 2017, but the actual first treatment was in animals in 2001. And so that's when we started working on trying to broaden the application. So this particular gene therapy is only for people who have one particular gene mutated called RPE65. And it's very few people. It's a very rare disease. And the virus that was used here only can remedy the RP65 deficiency. So again, very, very few people will be able to benefit from this therapy. But what it showed was that we could safely deliver a virus to the eye in a person and deliver basically a good copy of a bad gene and correct the disease. This has now been applied even to 2-year-old kids, and it's considered to be very safe and very efficacious. So this is our inspiration and the inspiration for many. So a little more background now. So when we talk about gene therapy, we consider a range of questions because not every disease is going to be dealt with by gene therapy. In fact, many, many diseases we won't be able to address with gene therapy. So the first question we ask is, what do we know about a disease? And the more we know, the more we'll know about whether we can add a gene or correct a gene to remedy the phenotype or the symptoms. And so we must have deep knowledge of the disease itself to consider it as a candidate for gene therapy. So that's very important. You know, there's a lot of diseases we have no idea how they come about. So it's hard to then address the cause, right? If you just don't know, why does a person get this disease? And there's many like that. So we would not be addressing those. The second thing we need to know is there a gene to remedy the problem? So again, if we understand what's going on in the disease, we can then think about, well, what kind of gene could we deliver that might correct things? So we have to have a handle on that. And then a very, very big issue is, how do we deliver the gene? And I'll be telling you more about that in detail, but I think one of the things that's often overlooked is, like, someone might understand the disease, they might say, well, I got the right gene, but if you can't deliver the gene to the target cells in the body that need the correction, it's not going to work. And so I consider, and as you see here in big letters, delivery, delivery, delivery to be crucial. The cells that you want to target have to be accessible to the treatment. And many types of cells in our body are buried in very complicated areas of the tissues, or they're very rare. And so those are actually hard to address with gene therapy. But some are very accessible, and those are the success stories. Okay, so how do we deliver genes? So the primary vehicle so far has been viruses. So probably most of you know that viruses are very small and they can't replicate on their own. They have to get into cells to supply all the components to replicate themselves. Many times they make us sick. They take over the cell's metabolism to make more of themselves, and the cells die or become abnormal. But some viruses are kind of innocuous. They can get into cells, they can replicate, but they do it so subtly that they don't make us sick. So one of the main viruses that we use is called the adeno-associated virus, and it's non-pathogenic, meaning it does not make us sick. So that one is a crucial one that we're using, and I'll show you examples. Now, some people, instead of using viruses, are using the bag of tricks that are developed by viruses for delivering their own genome, and they're making what are called nanoparticles. So nanoparticles are kind of virus wannabes, I call them. So they have components that are like viral components that enable them to deliver genetic cargo to cells when they're injected. And the most famous application, and one of the most early applications of nanoparticles, was for the COVID-19 vaccine. So I don't know how many of you followed this, but the company called Moderna, another one called Pfizer, they developed vaccines for SARS-CoV-2 by using nanoparticles. So inside a nanoparticle, they put a messenger RNA for a component of the SARS-CoV-2 virus. And that messenger RNA translated a protein called the S protein that's on the surface of the virus that enabled the immune system to recognize it as foreign. So it was a vaccine based on a nanoparticle. Other vaccines for SARS-CoV-2 use viruses, and those viruses have on their surface the same S protein to deliver to the immune system so that the immune system sees it as a foreign entity. So both have worked. The one from J&J is a virus-based one, and the one from Pfizer and the one from Moderna are nanoparticle-based. So there will be more such vaccines from nanoparticles and viruses. In fact, I believe one just got approved for respiratory syncytial virus, but this is an up-and-coming area where people are finding ways to deliver genetic material with nanoparticles that are designed for specificity. So far, they're not quite as good as viruses in terms of their ability to do that. But these are the two ways that we deliver genetic material for gene therapy. Okay, so how do we deliver genes using either nanoparticles or viruses? So there's two overarching strategies. One is to take cells out of the body. That's called ex vivo. You take the cells out of the body, you infect them, hopefully either deliver a good copy of a bad gene or correct, using something called gene editing, a bad copy. And then you put those cells back into the person. And I'll go over some examples of that. The other way to do this, and this was done for Luxturna, the eye disease I showed you, is to directly inoculate the virus into the tissue. So you'll see there's direct inoculations right into the eye. You don't have to take cells out and infect them and put them back in. In fact, you really can't do that for cells in the eye. You need to directly inject the virus. So I'm going to give you examples of both. So ex vivo. So the original target tissue for gene therapy, way back when I was a postdoc, so we're talking now 1980s, the idea was that bone marrow cells or blood cells are cells that we can take out of the body. You just bleed a person, take the blood, put it in a dish in the lab, and you can grow them. So that sounded like a good place to do gene therapy. So a little bit about this slide. So this is complicated, but this is a very simple version of what goes on to make blood cells. So in our bone marrow, so inside long bones, there's some cells that are called stem cells. These are called hematopoietic stem cells. And throughout our lives, they're dividing. And what they do is they make more of themselves, so we don't run out. And then they produce the cells that constitute our entire hematopoietic system. So those are our B cells and T cells and natural killer cells. So these are the immune cells that protect us from disease. So when you get a vaccine, for example, B cells and T cells are made that recognize the foreign entity. That's called the adaptive immune system. And so all those cells are continually produced by the bone marrow stem cells. There's other cells in the blood that have other jobs. Dendritic cells are cells that will gobble up viruses and then present pieces of the viruses to these T cells and B cells. So they're what are called antigen presenting cells. Other cells here, macrophages basically eat garbage. Dying cells, for example, are gobbled up by macrophages. Platelets, you may know, create clotting factors. So when you get a cut, you're bleeding, the platelets rush to the scene and they clog up the hole. So platelets are crucial for curing blood loss. Red blood cells, you probably have all heard of. They're super numerous in our blood and they carry oxygen throughout the body. And there'll be more diseases that affect those that I'm going to tell you about. So all of these cells are not that long-lived. They die and they're replaced by the hematopoietic stem cells. So the idea for gene therapy for hematopoietic diseases is, let's say you have a gene that's defective in the red blood cells. If you were to deliver a gene to the red blood cells, they die like every 80 days. So it wouldn't be a long-lasting cure if you delivered the gene just to the red blood cells. But if you deliver the gene to the stem cells, then it's a cure because they will be corrected forever. And all the red blood cells that will be created from those corrected cells will be corrected. So if you gave the gene here, eventually those cells die, treatment is over. If you give the gene here, it's permanent. So that's the strategy, is to give the corrective gene therapy to the stem cells, and then there'll be correction long-term for whatever cells are produced from those stem cells. So how does this actually work in process? So we have a patient who's got a disease that affects one of the blood cells. And so we have to get those stem cells out. This is the ex vivo idea. So to get the cells out, you have to stick a big needle into their long bones and suck out some of the bone marrow cells. That's one way. Another way is there's drugs that you can give the person, which are called mobilizing. And what happens is those bone marrow cells that are stem cells respond by dividing and then entering the bloodstream. So in that case, rather than sticking a needle in a big bone and sucking out bone marrow, you can just bleed the person and try to isolate the stem cells from the blood. So both things are being done. It sounds like a great idea to just take blood. The problem is the cells in the blood that are stem cells are a tiny percentage, less than 1%. So it's hard to get enough cells out of just bleeding the person, whereas you can get more if you just take it from the bone marrow. But either way, the goal is get out some stem cells, okay? And then in a dish in the lab, and I'm going to show you a movie where you see what they're actually doing. They try to enrich or isolate the stem cells by putting a protein on the surface that recognizes just the stem cells, and that turns the stem cells fluorescent. And then there's a machine you can put all the cells through, and the machine will pick out the fluorescent ones. So that's being done, and you'll again see a movie. Now, once you get the stem cells in a dish, you add a virus to do one of two things, and both things are being done successfully. So one thing is to add a good copy of a bad gene. And that's the simplest idea and the oldest idea. So let's say a person has a mutation in a gene that's needed in the red blood cells. And I'm going to show you an example. It's called beta globin. So if a person doesn't make enough good beta globin, you can put a good copy of beta globin in the virus, and then that virus will infect the stem cells, deliver the beta globin gene. And the crucial thing is this beta globin gene will integrate the DNA of the beta globin gene into the host chromosome. So that means that cell will forevermore have a good copy of the beta globin gene. And every time it divides, its progeny cells or its daughter cells will inherit the good copy of the beta globin gene. So again, it's a cure. It's a stable correction of the beta globin loss. So once that's done in the lab, the cells are put back into the person who donated the cells. What's great about that is when you put those cells back into the donor doesn't recognize the cells as foreign because it's their own cells. So you may have heard of grafts coming for other things, like someone might get a liver transplant, for example, or even a blood transfusion. And when you do that, you have to match the donor cells to the host so they don't get recognized as foreign or they get rejected. So rejection of grafts happens a lot. And that's because the cells you're putting into the person are not their own cells. This is called autologous grafts, which means the cells that you took from the person who has the disease, you fixed the cells in a dish and put them back into the same person. So there is no rejection. So this is a beautiful therapy for just stepping past, getting by the whole rejection thing. But there is some downsides. So to put the cells back into this person, usually their endogenous cells that are left are killed by a drug and radiation to create space for the engrafted cells to be able to survive and repopulate. So you want to get rid of the bad remaining cells. When you take cells out of the host, you're not removing all of the bad cells, just a portion of their bone marrow cells or their stem cells. There's a lot left. And so people are treated with radiation and drugs, which is very painful. It's also dangerous, to create space for the fixed cells to then survive. And so it's called myeloablation. And so it's not a great therapy from the patient's perspective in terms of the acute pain and agony they go through, but it's a long-term curative therapy, which again, there's good examples of now. So people go through this. Another downside is it costs $1 million. So in our country, so far, some of the insurance companies have been willing to pay for it, but it's obviously not going to be extrapolatable to everybody everywhere. But for some people, it's already shown absolutely amazing curative properties. Okay, so this is the ex vivo infection with re-engraftment. It's being done now for a couple of diseases I'll tell you about. But I'll also tell you one of the initial motivations was a disease that affected all of these cells. This was a disease called SCID. And if you've ever heard the story of the boy in the bubble, this might be too old for a lot of you, but this was a very popular story 20, 30 years ago. So SCID is a horrible disease where, again, all of those bone marrow derivatives are affected. And essentially, a kid that's born with SCID doesn't have an immune system. So they usually die of an infection by 1 year of age. So this boy, David, was born to a family in Texas, and they knew that their family had this disease gene because they previously had a child who did die in the first year of life. So when David was born, they were aware that he may also be affected by this disease, and they tested him. They found, yes, he has no good immune cells. So they thought they would find a bone marrow donor. So before gene therapy, the way that bone marrow diseases were treated is they would find a donor. Usually it was a family member that had similar looking cells to their own, so it reduced the chance of rejection of the graft. And then they would just take bone marrow cells from a donor person and put them into the patient. So they thought that David could survive if they could find a suitable donor. So while they were waiting, they didn't want him to get an infection. So they put him into a chamber called a bubble. And basically he was isolated from infections or the whole world. He lived in this plastic bubble, and all his food was sterilized, his clothes were sterilized, everything that was given to him in the bubble was sterile so he wouldn't get an infection. And the idea was that this would be a short time, maybe months, before they found a donor. And again, he was just a baby then. But they couldn't find a donor. It went on until he was about 10. He lived in this bubble. He eventually moved home, but what a horrible life. The poor kid lived in a bubble. So this family had a daughter after him, and the daughter was a good match. That is, her bone marrow could be accepted by David. So they had a good donor then. He went through the bone marrow transplant, and there was a tragic ending. The tragic ending was that the daughter's cells had a sleeping virus called a herpes virus. The people who did the graft didn't recognize that her cells had this herpes virus. She had no effect from the herpes virus. She was perfectly healthy. But once those cells went into David's system, the herpes virus reactivated and spread and killed David. So a horrible story, but it was part of the reason why people then went after SCID as the first gene therapy for bone marrow cells. And some of these trials have been successful. So the very disease he had has now been cured with gene therapy, because again, they can take out the patient's own cells, correct them, and put them back. So they don't have to wait around for a bone marrow donor. It's a good match. So SCID is being treated in kids successfully now. Okay, another big category, a much bigger category of diseases in the hematopoietic system are called hemoglobinopathies. So I told you that red cells, our red blood cells, carry oxygen all over the body, deliver it, and it does so by having a protein called beta globin. So there are two sort of big categories of beta globin disease. One's called sickle cell anemia, and one's called beta thalassemia. So in both cases, the red cells are affected, and the person doesn't get enough oxygen to their tissues. So the goal for gene therapy for the hemoglobinopathies is to supply more wild type or normal beta globin, or increase what's called fetal gamma globin. And this is a little bit more complicated. I'm just going to mention what it is, and then we'll see some movies of people that have been treated. But if you get lost in this next part, just take my word for it. There's basically two ways to fix things. One is to put in more what's called adult beta globin. That is putting in a good copy of a bad gene. The other way is to do what's called an editing, which is to create a mutation in the cells that gets rid of a repressor that keeps down the level of a different kind of globin called gamma globin, which takes the place of the beta globin. Okay, so here's the two strategies. And again, it's a little bit complicated. And if I lose you, don't worry about it. You'll see the big picture in a moment. But let's say a person's born with a mutation in their adult beta globin gene. So that then needs to have either a good copy added back, so it doesn't have this mutation, and leave this one in the genome. So the bad copy will still be there, but you can add back a good copy, and now you're going to have enough normal globin that you won't have lack of oxygen to your tissues. So that's a good copy of a bad gene. Another thing is when we're babies, when we're in the fetus, we have a different globin gene called gamma globin, and that copy goes away when we're born, and it's replaced by beta globin, the adult form. But we can reconstitute the expression of that gamma globin by getting rid of another gene, it's called BC11A, that represses the expression when we're born. So this is a repressor. Some of you who might know more about transcription might know that this thing is a transcriptional repressor. So the second way this is done is editing, which is a way to alter the genome, is to cut this gene so it's no longer functional. which means the gamma globin is going to be expressed in the adult. And this will take the place of the bad adult globin. So either we're doing something called CRISPR or gene editing, or we're doing something called gene augmentation. We're adding back a good copy of the beta globin. So again, 2 ways to do it. They both work. And now they're both FDA approved, which is very exciting. So here's just a picture of the editing. Just show you again that this BC11A protein is normally inhibiting the fetal globin. And you can get rid of this by putting in a CRISPR, an editing function that kills it. So I just want to show you a YouTube of a person that's been treated for this disease.
With sickle cell is understanding that, you know, some days I'm gonna have up days and some days I'm gonna have down. This is your body. This is what you use every day. Having sickle cell, you're not allowed to, I feel like, enjoy some of the little things that other people enjoy. I feel like obviously we've been dealt a crappy hand of cards, and you know the goal is to play to the best capabilities that you can.
Sickle cell anemia is a terrible disorder. It is a hereditary disorder. That is, it's something that occurs in one's gene, inherited from parents. Yeah, worldwide, it's the most common hereditary disease. So many millions worldwide. In Africa alone, 250,000 newborns are affected annually.
Actually, they're not the only ones. There's teams all over the world doing this.
So the hallmark of sickle cell disease is pain. It happens when, after the hemoglobin molecule, which is specifically designed to bring oxygen to our vital organs, delivers the oxygen, the sickle mutation, which is the change in the DNA, creates this long strand, making the round, flexible red blood cell into a very sticky, stiff, sickle-shaped, crescent-shaped red blood cell. And that blocks blood flow. So when you don't get enough oxygen to a tissue, causes pain. So patients who have sickle cell disease experience pain. They experience damage to vital organs where blood flow can be altered by the sickle red blood cells. Lung problems, when you don't get enough blood to your brain, causes a stroke. These are all common complications that affect the quality of life, not just the lifespan in a person with sickle cell disease.
I feel like I've experienced some, you know, really crazy amounts of pain. Like I've had both my lower extremities, you know, they had infarctions in them. So, you know, like imagine both your legs just being broken at the same time. It was the worst pain you can think of. And then my, like with my chest, my pericarditis hit, like I could barely breathe. I was in like constant pain all the time. So there was some days I just felt like a zombie, you know?
We know that if you correct that mutation, you take away the disease. So anyone could imagine that if it was possible to correct the gene in one's own cells, that you should be able to cure the disease. So when we first heard about CRISPR-Cas9, we got very excited because that could target the sickle gene, correct the problem, and then restore healthy red blood cells in the circulation. There are 3 groups of scientists and clinical investigators working together to accelerate the development of this technology in humans with sickle cell disease. So one group at UCLA will be involved with manufacturing, that is, producing the blood stem cells after correcting the sickle gene. And they are expert in that activity. The group here in Oakland are experts in hematology and sickle cell disease, and ultimately will lead a clinical trial when we begin to treat patients. Yeah. And then the anchor, I would say, is the IGI, the Innovative Genomics Institute at UC Berkeley, that really brought that technology to us. And they're leading the effort in documenting at the DNA level how many of these blood stem cells we've corrected. We don't have enough therapies for sickle cell disease. If it's related to societal pressures in underprivileged, underrepresented populations in the United States, how rewarding would be to deliver this amazing cure to this underserved population? It's justice, right? It's the first hereditary disease ever defined in human beings, primarily affects African Americans and underserved population, has difficult access to healthcare in some cases. And now, finally, we have a cure that we can offer the majority of patients, at least in our country. I mean, that would be terrific.
Okay. One more thing just to show you another correction. So people who have been treated by the gene editing, as I told you, it's a total cure. They go from going to the hospital once or twice a month, sometimes for 2 weeks at a time, terrible pain, and they die young. Those people now don't go to the hospital, don't have pain. It's an incredible thing. So that is an editing, and I wasn't planning to talk about editing, but I can certainly answer questions. But fundamentally, editing means you go into the DNA of a person and you fix something directly. The other way that's being treated, and this is also FDA approved, and here's a picture, is people are getting, again, a good beta globin gene delivered to their stem cells ex vivo, and then those stem cells are put back in. So now that person is making good beta globin. And here's just a picture before and after treatment. So on the left, these red arrows are showing us funny looking red blood cells, this sickle shape or just oddball shape. And then after correction, there's almost none. Here's one. So this is another one that's working really nicely. So this is kind of a miracle for people. I mean, in the long run, what we're going to need is drugs because, again, doing this kind of graft costs too much. And certainly in Africa, where most of the sickle cell patients live, they're not going to be able to have this kind of bone marrow transplant. But they will, if we could find a drug, and there are companies working pretty hard on finding drugs to correct the sickling. So that's one example of gene therapy that's been approved. Now I want to go to ocular gene therapy. And this is obviously what we're working on. And we're inspired by that first video I showed you where the first FDA approved therapy was carried out. And the eye has a lot of advantages for gene therapy. This is now for in vivo delivery. We don't take cells out and put them in a dish and put them back in, which immediately takes away a lot of the cost and the pain. This is very simple. A needle goes in the eye. I'll show you a picture and delivers a virus that carries a good copy of a bad gene or does editing. And I just want to point out, not only our eyes are beautiful, just for fun, I just wanted to show you this. They're incredibly variable, and this is something that we're fascinated by. And as we study how the eye develops, we consider how you get these different forms. But that's a topic for another day. Okay, here's just a diagram of a human eye. So I don't know how much you know about this, but obviously light's coming in through your pupil, and it's being directed to the back of your eye. And this yellow line represents what's called the retina. This is the nervous system of your eye. Again, this is actually part of the central nervous system or the brain. And this yellow line represents many cells, as I'll show you. So when we want to correct a retinal disease, we're going to try to infect cells here. And this black line represents another kind of cell called the retinal pigment epithelium. And this is just a simple epithelial sheet. And those cells supply functions to support the retina. And these so-called RPE cells are the target cells for Luxturna, the first approved gene therapy. Okay, so here's some of the cells in the retina in a simple cartoon. So light comes in over here on the left. You can see the light being represented by these arrows. It enters your eye through the pupil. It actually traverses all of these cells until it hits these photoreceptors. So this is where the light detectors are. This little one here is called a cone photoreceptor, and it's what you use for color and daylight vision. And the more numerous, actually, rod photoreceptors are here in purple. And they're what you use at night. So if you're walking around in the woods at night, you're using your rods. But all the rest of the time, if you see any color at all, you're using your cones. It turns out that most of the disease genes that lead to blindness directly affect rods and cones. So these are the cells we're always trying to save. Here's a real picture. So these green structures, like let's look at this one. This is a rod. It's called a rod because it looks like a rod. And this part of the cell is where light is captured. And then there's a biochemical process called phototransduction that allows the rod cell to change the output of a neurotransmitter called glutamate. And these other cells down here, so here's where glutamate is released. So here's where light is captured. And then a neurotransmitter called glutamate, a small molecule, gets released. And then all these other neurons in the eye process the signal. So the eye's not a camera. This is kind of like a camera, but it's a parallel processing computer. So all of these cells down here are analyzing the input of light as directed by the photoreceptors. So some of the circuits that are the parallel processing circuits are analyzing color, some are analyzing motion, some are analyzing edges, some are analyzing changes in illumination. We probably have about 40 circuits that are being used right in your eye, not in your brain, but right in your eye to analyze the pattern of images that are falling on your retina. And then after all that information processing takes place locally, these are called axons. These are very thin processes that go to the brain to tell the brain what the retina just processed. And then in your brain, you recognize things. So like the retina processes different wavelengths of light, which gives information to your brain to say, oh, that's the color purple. Or your brain says, oh, that's my mom, right? It can recognize a shape, and there's a stored image that allows you to recognize anything. So, but local processing with all of these neurons, and there's about 100 types of neurons here, creates information for the brain to work on. But most of the time, the disease genes are just affecting the rods and cones. So again, that's our main goal. So it turns out there's a lot of genes that lead to loss of vision. The most numerous of all human disease genes affect vision. And this is just a little pie chart showing you the number of genes that affect the ear and cause deafness, hematology, again, the diseases we just talked about, the heart and blood vessels. But if you see here, and this isn't outdated, there's more now, 647 disease genes can lead to loss of vision. So there's a lot of them. And the reason we probably know there's so many is because we are super visual. So if a person can't see color or has blurry vision, they immediately go to the eye doctor. Whereas if you have, let's say, something that affects your blood vessels, it might be more subtle. You might not know you have a disease, but as soon as something affects your vision, you know it. And that's because we are totally dependent on our vision. So that probably helps with the genetic diagnosis. So human geneticists have lots of people they can analyze for disease genes because we self-report. So at any rate, there's so many of them that we think we're not going to be able to address each disease gene by adding a good copy of a bad gene or by doing editing, because each of these therapies to develop for a single gene costs millions of dollars. And companies are just not going to do it for 600 or so genes. They'd have to have a different clinical trial for every one. It's going to run them broke because there aren't enough people that are affected by any one of these to give them a profit. So our strategy is trying to do something generic, something that'll help many different disease gene families without being specific. And essentially what we're doing is trying to make the cone photoreceptors, the ones we use for color, trying to make them more resilient in the face of sort of bad genetics. And I'll tell you more about that now. So one of the categories of diseases that leads to blindness is called retinitis pigmentosa. And 1 in 3,000 people in our country are born with it. So it's inherited. You get it from your parents. And it has an early to late onset of loss of vision. I'll show you some examples. And that depends on what exact disease gene. There are many different disease genes. I think we're up to 100 different genes, when they have a mutation, lead to this retinitis pigmentosa disease. Now, many of these genes, so you probably all know that every cell in our body has the same genes, but different cells turn on different genes, and that's why we have different cell types. So red blood cells, for example, make beta globin. No other cell makes beta globin, but all the cells have the gene for beta globin. Here, for retinitis pigmentosa, most of the disease genes are only expressed in the rod photoreceptors. They're not expressed in the cones or any other cell type. They're rod specific. So that explains the symptoms. So what happens is a person is born night blind. Now, because we have electricity now, a parent might not even know that their kid is night blind until they, let's say, run into something in the backyard at night. or fall into something, you know, so something that relies on the rod vision, these rod photoreceptors, which have the disease gene or express the disease gene, that's when a person knows that they have this disease. And again, because of electricity, if you think about it, most of the time you see color, even at night, you see color. And that's because you're using your cones, and your cones don't express the disease gene. But as soon as you're relying on your rods, let's say walking in the woods at night, you will not be able to see where you're going. And that's how you know you have this disease. So what happens over time is that those rods die, and it ends up then creating what's called tunnel vision. So right in the very middle of our eye are no rods, only cones. That's where we have actually our high acuity color vision. And eventually those cells die, and you can become totally blind. So here's just what it looks like. So a person is born with normal color vision. And so every time there's enough light, the cones are active. They don't know they have a rod disease. But over time, the rods die. And the key thing about what I'm going to tell you is when the rods die, then the cones that are in their neighborhood start to dysfunction. So that means you start to lose color vision. And first you lose your color vision more peripheral. And then eventually you can lose all of your cones and your rods. You can become completely blind. And there's no treatment for this disease. So there's a second disease I'm going to tell you about, which is really common in the elderly. It's called age-related macular degeneration, or AMD. And these people have the opposite. So instead of having their peripheral vision first affected, their central vision, which again is our high acuity daylight vision, that's affected. And this one affects 25% of people in our country, age 75 or older. I don't know if any of you have grandparents that might have this, but it's pretty common. Now, this is different than retinitis pigmentosa. So in retinitis pigmentosa, you're born with a single disease gene. And if you have that disease gene, you are going to get the disease. This disease, age-related macular degeneration, we don't have what do we call these deterministic genes. Instead, we just have certain mutations that give you a higher probability of getting this disease. So it's not like one gene defect gives you the disease. It's versions of several different genes that put you at higher probability to get it. And these are called risk factors, genetic risk factors. And what that means is, as I told you at the very beginning, we don't really understand why people get this disease. You know, in the case of retinitis pigmentosa, we can identify the disease gene. We can say, well, what does that gene do in the normal vision? And we get some understanding of why a person's rods die. Here, we don't have that advantage. We don't really understand the disease. We know there's risk factors, and those risk factors are many different genes. They affect many different pathways. Most of those pathways affect all cells in the body. So why is a person just losing their high acuity central vision? We don't know. So in this case, what our lab is trying to do is to treat some of the symptoms, and I'll tell you about that. We're not sure that's going to be successful. Now, there's two forms. One is called wet, one is called dry. You may have heard of the wet form. The wet form is treatable by eye injections with an antibody that I'm not going to go into. The dry form is far more prevalent, and we don't have a treatment. So that's the one we're trying to target. Okay, so this is the opposite. So a person who has macular degeneration now loses their high acuity central vision. So they have some peripheral vision, but that means they have to move their head around, their eye around, trying to capture whatever they want to look at. Naturally, we're directing the light right into the center, but that's where they don't have vision. They can also lose all their vision. Okay, so that's the two diseases I'm going to talk about: retinitis pigmentosa and age-related macular degeneration. Okay, so how does this look if a person, an ophthalmologist looks in the eye of a diseased person? So this is the retinitis pigmentosa eye. And if you go to the eye doctor, you know, they shine a light and they look in your eye, and here's what they see if you're normal. So these are the blood vessels that nourish all those retinal cells I showed you. They come in through something called the optic nerve head. Right here in the middle, this dark spot is your high acuity vision. This is the macular region, which is affected in the age-related macular degeneration. So when you normally, with normal vision, what you're doing all the time during the day is you're trying to get whatever you're looking at for those light rays to fall right here, not out here, but right here. So that's why we move our eyes in our head, or we move our head. So the light, like when you're looking at a screen, you're looking at directly at the screen, like your eyes are pointed right at what you want to attend to. And that's so that the light hits right in the center. And you'll notice, do a little experiment with you. If you hold your hand up right in front of your face and you look at your hand, you see every detail, right? You see your fingers, you see all kinds of interesting details. So now if you keep looking straight ahead and you move your hand to the left or right, you can't see any of those details. You can see if you move your hand, there's movement over there, but you lose all details. Okay, so that's the difference between your peripheral and your central vision. Your central vision is high acuity and your peripheral vision is not. Okay, so that's normal. Now, if you have the disease retinitis pigmentosa, it looks awful. And you can see where it gets the name. So retinitis basically means you have an inflamed retina. You've lost a lot of the nice blood vessels. So all these beautiful blood vessels are going away. And that's because you have something called hyperoxic eye, means you have too much oxygen in your eye. And I'll explain why in a moment. And when you have too much oxygen, the blood vessels, which deliver oxygen, sense that and they regress. So you can see this person doesn't have many blood vessels. The second obvious thing is all these black cells. I told you that there's these support cells called RPE cells. And when a person has retinitis pigmentosa, those RPE cells, which are behind this image, they migrate into the retina, which they should never do. So that's where the name pigmentosa comes from. So at this stage in the disease, a person has lost their peripheral vision, both rods and cones. They can't see at night, they can't see in the day in their periphery. They're getting this weird inflammation in the center, so they're starting to lose their high acuity central vision also. And again, this can progress to total blindness. So this is a disease that we're targeting. All right, just to show you what the tissue looks like. So the view I just showed you was a doctor's view, an ophthalmologist view. In the lab, what we do is we have mice that have the same disease, retinitis pigmentosa, as people. They have the same disease gene, same mutation as people. And we use that as a model to test our therapies. So what I'm showing you is a cross section. So we took an eye out of a mouse and we cut a section right through it. This is like the cartoon picture I showed you. So here, all these blue dots out here, every blue dot is the nucleus of a rod. And up here are the nuclei of cones. Now in our eye, 95% of the photoreceptors are rods. That's why we have great night vision. 5% are cones. Normally in the periphery, which I just told you how you have low acuity in the periphery, that's because in the periphery, we only have 5% cones. So there's not many cones. And right now we're in daylight. So that's why you couldn't see the details of your hand when you put it to the side. There are so few cones, only 5% of your photoreceptors. But right in the center where I told you you have your high acuity vision, we have only cones. They're packed in, super dense. And so in the daylight, you get really high acuity vision. You can imagine it as sort of a high pixel density receptive field. So in the mouse, which we use for our model, there is no high acuity central vision, sadly. And all mammals don't have central high acuity vision except for primates. And obviously we're primates. So we have central high acuity vision because we have these cones all packed in the center, but mice don't. So what we're doing here is we're just looking at what it looks like in the peripheral human retina, which is tons of rods. So all these little blue dots out here, each of these is a rod. All these other blue dots belong to the interneurons that I told you carry out the circuitry for extracting information for vision. So we're going to ignore those for now. So all these rods are diseased in the model we're looking at here. They're not yet dead, but this mouse can't see at night. These red structures out here are stained with something called PNA, and it allows us to see the cone outer segments. Again, in the early slides, I told you the outer segment is where light is captured and phototransduction takes place. So in this stage, in this disease model, the cones are healthy. You see all of these nice outer segments. So that means this mouse can see color during the day. And the rods are still here. Now, the next slide I'm going to show you, the rods all die. And secondary to rod death, the cones start to degenerate too. But, again, they don't express the disease gene. So they're dying because the rods are dying, not because of intrinsic genetic problems. So this mouse we looked at 8 weeks of age, and then the disease progresses over time to the loss of rods. So now this is 17 weeks that we're left with a single row, 1 or 2 rows, which are rods and cones. The cones haven't died yet, but now their nice outer segment that captures light has collapsed. So at this stage, the mouse is losing its color vision. Same in a person. So they won't be able to see at night from birth because their rods don't work. The rods die. And then because the rods die, the cones get sick and die. So that means there's a bystander effect. Something happens to the cones, not because of genetic problems, but because their neighbors died. And that's what we're trying to address, because we think that might be a common set of problems across all different disease gene families that have retinitis pigmentosa. So we want to know, if that's a common problem, can we save the cones across different disease gene families? But what genes do we put in? How do we save them? First question. And that comes back to what I told you in the very beginning. We need to understand the disease etiology in order to figure out what genes do we put in. Second question, how do we introduce the genes? And then, what kind of genes, how many? So first, how do we introduce them? So this is the virus that is used for Luxturna. I showed you Corey Haas, the boy in the first video. He got AAV gene therapy. That means the virus, adeno-associated virus, was used to carry in a good copy of his bad gene. It's also the virus that's being used for a lot of the editing and other diseases. So it's the most popular gene therapy vector. And the reason is it's not pathogenic. It doesn't cause disease, and we can manipulate in the lab pretty easily. So this virus has its normal genes removed, and then its DNA, its genome, is manipulated so it expresses, let's say, a rod gene or a cone gene, and it uses a transcriptional element, something that turns the gene on only in the right cells, called a promoter. So we engineered that in the lab, and then it's a simple injection. I mean, it looks like it's painful, but it isn't. The person's eye gets anesthetized, and this can be done in the doctor's office. Right now it's being done in the hospital just as a precaution, but in the long run, it can be done in a doctor's office. So we have a very thin needle, and we inject between the retina, the photoreceptor side, and this RPE support layer I told you about. So the virus goes into what's called a bleb. It doesn't flow throughout the eye in a person. In the mouse, we're treating an eye that's 20 times smaller, and the virus goes everywhere. So we're infecting all the photoreceptors and all the RPE cells. In a person, in reality, we're infecting only about 15% of the cells we want to treat. And that's because the human eye is so big. So this is one inoculation. So that's it for life. You get one inoculation, and the virus then infects the cells right at that bleb site. So the RPE cells get infected, the photoreceptor cells get infected, and then the hope is that the cells will live longer. Okay, so just to give you a little bit more about AAV, some of you may have heard of parvoviruses. If you have a dog, there's something called canine parvovirus that dogs get and makes them sick. But again, when we get infected with our AV, it doesn't make us sick. And there's a few little details here. Actually, I think I'll skip them. It's not important. But we wanted to test in our mice whether we could get the photoreceptors infected, infect the RPE cells, and then turn a gene on after the infection. If we couldn't do that, we couldn't move forward with our therapies. So first we had to test, can we infect the cells? And then once the DNA from the virus is in the cells, will it turn the therapeutic gene on? And as a proxy for the therapeutic gene, we put in a fluorescent protein gene called GFP or RFP, which turns the cells green or red. And that's an easy way for us to assess, did it work? So these are now cross sections from eyes that we infected. So here we have a cross section, and what you see with green is the viral gene being expressed. This is called GFP for green fluorescent protein for obvious reasons. Each of these is a cone cell. So here's the nucleus. Here's the axonal ending where the glutamate is released. In red is this outer segment where light is captured and phototransduction takes place. So this is a normal eye, and we had beautiful full infection. All the cones got infected. They all express a disease gene. So this is a great result for us. If we do this in a diseased eye, where I told you all the rods are dying and there's just some cones left, it still works. We can get diseased cones infected. So this means we can try to treat the diseased cones. Okay, so how do we do the experiment? So in the lab, we have 3 different strains of mice that have the disease, the retinitis pigmentosa disease. That means we now have a model organism or model disease system where we can test for therapeutic effects. And that's actually crucial. There's a lot of diseases where we don't have a good model organism. Like I told you, AMD, we don't have an animal model for the macula because mice don't have the macula, rats don't have the macula, fish don't have the macula. Lots of animals don't have a macula. But again, among mammals, it's just primates, and those are not easy to use for experiments. So we're not using monkeys right now. So as a proxy, though, we use for retinitis pigmentosa, it's not a macular disease, it's the whole eye. So we can use mice and rats pretty easily. And I'll show you as a proxy, we can also use retinitis pigmentosa mice for AMD treatments. But here's what we do. So we take viruses and we engineer them. So one virus carries a green fluorescent protein that will turn on only in the cones. And what that does is it gives us an easy assay. We can easily count the cones to see if we save them. We can also put in other genes that are potentially therapeutic. Now, one problem with these AAV viruses is the genome is small. We can't put in multiple genes into the same virus. So sometimes if we want to deliver more than one gene, we make more than one therapeutic virus and we co-infect with all of them. And we can do that pretty easily in a mouse. We can just inject a lot of virus and all the cones get co-infected with 2, 3, 4, 5 viral particles at the same time. So after we inoculate, we do this at postnatal day 0 when the mouse is born because it allows the inoculum to flow all through the eye. And as I showed you with the human case, if we inject an adult, for anatomical reasons, the virus doesn't flow everywhere. But in the mouse at postnatal day 0, we get a flow of the virus and we get all the cells infected. So that's why we do postnatal day 0. And then we let the animal grow until some stage in the disease process. So our 3 disease models, some lose vision quickly, some are slow. When we're ready to test, we either do an assay for vision called the optomotor test, which is down here on the left. So we put the animal in what looks like a rotating drum of stripes. And this is an interesting thing that all eyes do. So if we're presented with a moving stimulus anywhere, we involuntarily move our eyes to follow the movement of the stimulus. So we can put these stripes, these black and white stripes, to simulate movement. And we can test by asking, does the animal move its eye when we simulate movement of these stripes? And if it does move its eye, it means it has vision. And we could also say, well, how good is the vision? So we can make the stripes really fat or really narrow. And by varying the width systematically, we can get a measurement of the acuity of the animal. So this is our way to say, when we added a therapeutic virus, did we extend the vision of the mouse? We also can count the cones. So we take out the eye, which is sort of like a hemispheric ball, and we make some cuts so that we can flatten it out. And then we can count how many cones are left by either counting the green cells, which are from this virus infection, or we use antibodies that will mark the cones. So we can say, did we save the cones with our treatment? Now, what genes do we introduce? So this is just a little cartoon telling you the disease process again. So remember, the disease gene is only expressed, that means it only makes its RNA and protein in rods. So the person is born with no night vision because we need rods for night vision. Then the rods die. That's what this red curve represents. And then the cones start to get sick and the person starts to lose their daylight vision. And again, this happens across different disease gene families. So it's a common progression in many, many different disease genes. So that's why we're thinking there's a common pathway that leads to cone problems. So what are those problems? Why do the cones die? Just because their neighbors died? Well, maybe the rods make some type of growth factor or positive factor that the cones like and they need to live. Or maybe as the rods die, they release a toxin. Or we know from measurements that when the rods die, and remember they're the major cell type for the photoreceptors, the oxygen, which is normally coming into the eye, comes in at a very high rate in a normal eye. But when the rods die, the oxygen keeps coming in at a high rate. It doesn't get dialed down, which means the remaining cones are bombarded by oxygen. And if you're bombarded by oxygen, it creates degradation of proteins and nucleic acids and lipids. It's called oxidative stress or oxidative damage. So we know that happens in these diseases. You can measure oxidized proteins and lipids. So oxidative stress is definitely happening. We discovered that there's also metabolic problems that I'll tell you about. In short, it looks like the cones are not getting enough glucose, and they need glucose to fuel their metabolism. And lastly, in these diseases, and pretty much any disease in any organ, there's inflammation, which means the blood cells are reactive to tissue damage or some other kind of problem. And sometimes they come into a damaged tissue and they inappropriately do various things that are negative. So you know when you get a cut and it swells up, that's inflammation. That's a bunch of cells coming in and releasing contents. And in a good case, they clean up the garbage and they leave. In the bad case, they stay there and they create problems. So that's inflammation, and it happens in lots of diseases. And we know it's happening in these eye diseases. So these are correlated things. These are things we see, except maybe the trophic factor idea, but we see these things. And now the question is, did they cause the cone death, or are they just correlated? And the way we're approaching that is to say, well, if we address one of these problems and the cones live longer, it means that they are contributing to the death of cones. So we're doing these one by one. So what we hope to do in a person and in our mice is when a person shows up at the eye doctor and they know they're losing cone vision, that is their daylight color vision, at that point, we know they've lost most of their rods. And now we want to protect the remaining cones to extend their vision. We're probably not going to cure it, but we're hoping to extend their color daylight vision for a number of years. So oxidative stress is what we dealt with first. So what is it? Again, it's too much oxygen, and also the eye is exposed to light. So high light and high oxygen creates what's called free radicals. These are oxygen molecules that have only one electron in their outer shell, and molecules like to have two paired electrons. And so these oxygen molecules run around and they steal oxygen or electrons from other molecules like lipids, DNA, proteins. And when they do that, they create bad outcomes. Like they damage the DNA so it's mutated, et cetera. So oxidative stress is happening due to these free radicals. And as I've drawn here, there's oxidative stress in retinitis pigmentosa, and AMD, glaucoma, which is death of a different cell type, Alzheimer's disease, Parkinson's disease, many diseases have oxidative stress. So now our question is this contributing to the death of the cones? And how do we address it therapeutically? So what we do is we put in genes that encode two enzymes that degrade or blunt the free radicals of oxygen. So ROS means reactive oxygen species. And these two genes, SOD2 and catalase, destroy the free radicals. So we said, oh, if we put in the genes with our AAV viruses for SOD2 and catalase and we destroy these free radicals, do the cones live longer? And the answer was yes. So now this is a flat mount, we call it. So we've taken the retina out, we made cuts, we flattened it. And every little green dot is a residual, still alive cone in one of our disease models. And what you can see in the center of the retina, there's fewer green dots. In the periphery, there's more. And that's how the disease normally progresses in a mouse. Cones in the center die first, cones in the periphery die last. And so this is a control. So this is a mouse that was not treated with our disease therapy. It was just injected with a virus that has a green fluorescent protein. So it's a control. It should be neutral. And this shows us how many cones are left at this time in the disease. So the other eye in this mouse was treated with these antioxidation genes. And you can see we have lots more cones. So this was great, but it kind of wore off. So it lasted for a few weeks and then more cones died. So we said, well, it's good, but not good enough. So how can we make it last longer? Maybe also, can we even create more antioxidation gene therapy by putting in a different gene, a transcription factor? So what is a transcription factor? I hope some of you know this. These are proteins that bind to specific sequences on a DNA molecule, and they turn on genes. So one of them, called NRF2, turns on a bunch of different genes, SOD2 and catalase, which we already tried, plus more genes that fight oxidative damage. Plus, NRF2 turns on genes that fight inflammation. It turns on hundreds of genes. So does another transcription factor called PTC1 alpha. So we thought, okay, we're on the right track. Fighting oxidative damage makes the cones live longer, but we want to extend the effect. So let's put in NRF2 or PGC1 alpha or both and see if they make the cones live longer. So we did that and it works. We get even longer effects. Oh, I took out the picture that shows you the quantitation, but it lasts at least twice as long as the SOD2 and catalase. It does eventually wear off, but it does last longer. Okay, so then we wondered, did we even create what we thought we would, which is less free radicals, less oxidative damage? So this is just one bit of data to show you. So again, we're cutting a section through the eye of a diseased mouse, and on the left is the control eye. So we infected with just GFP, no transcription factor. Green shows us where the virus landed. Those are all the rods and cones that are shown in green. The same eye we tested for free radicals by putting in a dye. And the dye, when a free radical binds to it, turns bright red. So you can see there's lots of oxidation going on here in this control eye. But if we put in GFP plus the NRF2, and here's the infected cells, we've greatly reduced the amount of free radicals. So this is probably why the cones are living longer. We did other tests. All the tests tell us, yes, we're reducing oxidized proteins, oxidized lipids, and oxidized nucleic acid. So the therapy is working the way we were hoping, and we tested it in 3 different models of retinitis pigmentosa. It worked in all of them, which means it might be generic. It might work for more than one disease gene family in people. That's what our hope is. Oh, one more picture to show you some data. So I told you that these outer segments of cones is where phototransduction takes place, and they degenerate as soon as the rods are gone. So this is now a picture showing us in a control infected eye. Each of these green cells is a cone, and we stained in white for the outer segment. And you can see there really aren't any outer segments in this control. But if we put in the NRF2, now these green dots show us that we're regenerating the outer segments, which is probably why the animal can see better. We put it through the optomotor test, and we know these animals can see better. So this is also a good result. Okay, so then we thought, well, okay, we're hoping this is going to work for retinitis pigmentosa. Will it also work for AMD? Now, again, we don't understand AMD, but we know AMD has oxidative damage. You can measure, again, oxidized proteins and lipids, and for sure, AMD patients have oxidative damage in their eyes. Now, again, just to tell you, this is very prevalent. And again, because it's a disease of the elderly, we think that it's just going to keep increasing in prevalence. It's very expensive. There's a lot of visits to the eye doctor. There's a lots of loss of work years for people who are affected. And it costs a lot eventually to the company or to the country, but it also costs in terms of quality of life. And we have no treatment for the major form. Now, people think that the oxidative damage is part of the cause. We're not sure, but that's one of the best hypotheses we have. And so we thought, well, we have now a virus that fights oxidative damage. Could it help with some of the cells that die in AMD? So in AMD, the first cell type that ophthalmologists see as being affected is these retinal pigment epithelial cells. Oh, I just want to show you one more picture. This is what the ophthalmologist sees when he looks at AMD. So here is, again, a normal eye. You see all these beautiful blood vessels, and there's the macula. But in AMD, now what you see is these yellow spots. And if you took a cross section, what you see is gunk. This is degraded proteins, lipids, accumulated as a lump right underneath between the RPE and support cells for the RPE blood vessels. It's called drusen. And so if a person comes to the eye doctor and they have this, usually they're going to lose their central vision. It progresses over the years, and eventually the RPE cells die, and then the photoreceptors die. So we're seeing if we can deliver our antioxidative damage therapy to RPE cells, might we slow down this disease? Since we don't have an animal model for AMD, we decided to use our retinitis pigmentosa models because they also have death of RPE cells and they also have oxidative damage. And just to show you the interaction here in this cartoon, this is a cross-section cartoon. This is a single RPE cell. And what the RPE cells have are what are called apical processes. And they surround these outer segments of the rods and cones. And there's waste removal. So the photoreceptors have waste. They bud off little vesicles that are taken up by the RPE cells and then degraded in the RPE cells. The RPE cells deliver nutrients. So outside of the RPE cells is a rich plexus of blood vessels. And the blood vessels give oxygen to the RPE, glucose and other nutrients. And those nutrients travel through the RPE and get delivered to the photoreceptors. So now if the RPE cells are sick, they might not be able to carry out these functions. And that might be why the photoreceptors die in AMD. But again, we didn't have a model. We wanted to know, does the RPE cells get sick and do they have oxidative damage in our retinitis pigmentosa mouse models? And the answer was yes. So we thought, well, could we protect RPE cells with our NRF2 therapy? This is a different, there we go. So if we look at a sheet of RPE cells, so en face, if we rip out the RPE, flatten it out as a sheet and stain it with an antibody that shows us their outline, each of these little hexagons is an en face view of an RPE cell. So you can see in a normal mouse, these are beautiful. They're very regular, they're hexagonal, they're carrying out their function. But in a mouse that has retinitis pigmentosa, now you can see these RPE cells are very abnormally shaped and they're dying. So we thought, okay, now we have an assay. We can test whether the NRF2 will protect those cells. And so we put in our NRF2 virus. And what we got was rescue. So let's just look over here at the bottom right. This is a diseased eye, same stage as the one above it, but now it's got NRF2. And you can see it's got the beautiful hexagonal morphology. So we rescued it. The other thing is we looked with electron microscopy, which gives us a very high magnification view. And now we're looking at the inside surface. I showed you those microvilli in the cartoon. That's what we're looking at here. Each of these little structures is a microvillus that would be surrounding and supplying a photoreceptor with nutrients and taking away waste. So that's what it looks like in a normal, healthy eye. We've lost those cells in the disease. They're gone. The microvilli are gone. And then with the NRF2, we restore that. So we think now this might be a good treatment for AMD, and we're working with a company called Spark Therapeutics to bring this to a clinic, test it out on humans. Okay, to summarize this, we have the NRF2, which is a transcription factor, which is fighting oxidative damage. It also fights inflammation. It changes some genes that have to do with metabolism. And we can deliver it with our AAV and have it turn on only in the RPE or only on in the cones. And so we're going forward with that. Okay. I don't know when I should stop. Sean, I can stop now or tell you a little bit more about some other therapies. I want to make sure we have time for questions. Sean, stop or go on?
Yeah, I think now might be a good stopping place. The microglia astrocyte stuff looks cool though. Maybe if people want to stick around after and you have time, we can talk about that.
Okay.
But
Sounds
we can
good.
open up to questions for now.
Okay, I'll stop sharing and hopefully some of you will have questions.
I have a question.
Sure.
So like you said that because when the rods die, there's excess oxygen in the eye that leads to the cones dying as well. Like,
Yes.
shouldn't the nervous system know that if the rods die, there should be a lesser amount of oxygen going there, like the parasympathetic division? Why is it that the same amount of oxygen is still going and they don't get that signal that there should be a lesser amount?
That is a very, very good question, and there's no answer. I thought the same thing. Like, why doesn't the oxygen just get reduced in terms of delivery? I don't know why.
Okay.
Yeah, be a good way to. In fact, we've been trying to think of ways to turn it down. We haven't come up with any good answers. Great question. Anybody
Thank you.
else?
Wait, I have a question.
Sure.
So I remember at the beginning of the lecture, you mentioned that we're using like nanoparticles for some vaccines. So I wanted to ask, like, why have we been looking for alternatives instead of viruses? Like, are there any downsides to using them?
Oh, that's a great question too. So a couple things. Sometimes we make vaccines in viruses and they just don't work. We don't know why. Again, it's kind of crazy. So people have been trying to make an HIV vaccine for a long time, a malaria vaccine for a long time, and they're using viruses to deliver some of these things and they don't work. Now we have some ideas about the HIV one, but it's really hard. It's really just hard to know for sure. So people start looking for, well, should we be delivering the antigen, the thing that we're trying to get the immune system to recognize, using some other modality? And so they've thought about nanoparticles. The other thing I'll say about nanoparticles, which is better than viruses, is that they're really easy to re-engineer. So again, I'll go to Moderna. So Moderna has a particle and they put a messenger RNA in for the spike protein. And it's trivial to change the spike protein sequence according to the way the pandemic is proceeding. So you may know that as the pandemic goes, we get new versions of SARS-CoV-2, and some of the initial vaccinations create antibodies that don't recognize the new versions, the evolved versions of the spike protein. So that's why we keep getting revaccinated. And so it's easy to just change the sequence to the newest version of the evolved SARS-CoV-2 and put that new genetic material inside to a nanoparticle that can take place in days. Whereas if we have to re-engineer a virus and test it, it takes weeks to months. So it's very fast as a platform to introduce changes for the vaccine. So that's one advantage of the nanoparticle. A downside of the nanoparticle, and you may also know this, is it needs cold storage. So the viruses can be shipped at room temperature, especially to under-resourced countries where they don't have -80 freezers. So the nanoparticles need to be kept at -80. That is only in very rare places, like it's in hospitals, it's in labs, but it's not a common thing. So distributing the nanoparticles is not good worldwide. So that's a downside of nanoparticles, which is why we have both going on. And in our country, we actually use both for populations, and they both work. In some other cases, it might be one works better than the others. It's very idiosyncratic. One works better for one disease, the other works better for another disease. We don't really know why most of the time.
Thank you.
Sure. Okay, I see lots of questions. Go ahead, anybody.
All right, so how much would this therapy cost? The one for retinitis and AMD.
Too much, really too much. So the companies that have developed these technologies have spent years developing it. So they want to recover those costs. So $500,000 for one treatment. Now they justify that by saying, first of all, we spent a lot of money to get here, so we want to recoup those costs. That's one reason. The second reason is if you look over the lifetime of a person that has some of these diseases, there are a lot of costs associated with the diseases. The person has lost their ability to work, and people put a number on that. Quality of life, I'm not sure how you put a number on that, but people do. So they say, well, if you look across the decades that a person would live when they're blind and all the medical system attention to that person, it's less if we give them gene therapy. Okay, the cost is less. So they justify the high cost by saying we're saving money. over that person's lifetime if we actually cure them. But it is 500,000 to 1 million for a lot of these gene therapies. All of those graphs that I showed you for sickle cell, et cetera, those are more like 1 million because they do have a lot more costs associated with the hospitalization. These myeloablation therapies, everything is just costs more. The AAV therapy where you just inject it in the eye is simple. Like I said, it's eventually not even going to take hospitalizations. It's just going to be a doctor's office visit. And once they make the AAV, it's really easy to store in the freezer forever. So the manufacturing, once they get there, isn't that big a deal. So I hope the costs come down for those treatments, but right now it's 500,000.
Thank you.
Sure. Anybody else? Don't be shy.
I kind of have like two questions.
Yeah.
So like for my first question, I wanted to know, you mentioned the sickle cell disease and how it's very common in in Africa. So one thing that I kind of heard about, what I wasn't sure, is that the sickle cell disease, if you have one gene copy of it, makes you like immune to malaria. So is that one of the reasons why these kind of diseases kind of spread? Because even though they're causing like generally bad things, they do make you immune to certain diseases.
Absolutely, that's exactly the right thing. That heterozygous, so one copy, it doesn't make you immune, but you're protected. You'll live longer and better if you get a malaria than a person who doesn't. So there's a selective pressure for it to spread through Africa where malaria is high incidence.
Yeah, and also like what are like a few examples of like new scientific like foundations or technology that you think is going to be a big part of like our future in the next few years?
Yeah, so I think gene editing, which I only touched on, where one can go in and change the DNA directly, that is exploding as a technology. And there's many different modalities. Part of the reason I didn't get into it is we have base editors, prime editor, Cas9 cutting, RNA editing, RNA destruction. Like there's all these new modalities that are being developed technically. And the challenge isn't now, those therapies are getting to the point where they're effective. The problem is the delivery. I mentioned at the very beginning, the delivery. And so the sort of frontier there is to get the delivery to the cells you want to correct, but not have the editing machinery hang around too long. So just long enough to affect enough correction in enough cells to get therapeutic effect. But if you leave editing machinery in cells forever, which is what we do with AAV, it might have off-target effects. It might cut DNA or RNA where it should not. So delivery of gene editing and regulating gene editing with all these different modalities is very much at the forefront of research right now. And going forward, I think that's going to be a big deal for correcting genetic disease.
All right, yeah, thank you.
Sure. Anybody else?
Yeah, I had a question. My question was, so I'm doing a lot of AI research in this program. So I wanted to ask, how do you use AI in your lab? And if so, like if you're going to use it in the future, where would you use it?
Yes, so we just started to use AI. We haven't gotten too involved yet, but if you're in AI, you may know the program called AlphaFold 2. You know about that? Do people hear that? So this was a huge breakthrough that was enabled by two things. One is computational power, and the other is the data that scientists have collected over decades now, where they crystallize proteins and they look at the structure of those proteins. And like when I was a grad student, so we're talking back in the late 70s, if someone solved the structure of a protein, it would be on the cover of a magazine. It was like a really big deal. Now there are hundreds or thousands maybe of protein structures that have been solved. And those were fed into a computational pipeline where the program would look at the primary amino acid sequence and the three-dimensional structure of the protein and try to understand how does the primary sequence of amino acids lead to that 3D structure. This has been the frontier in protein biochemistry for decades. And the breakthrough came because of the computational power again, and simply the number of examples of crystal structures we have of proteins. Those structures were used as training for the AlphaFold 2 to be able to then, given a primary amino acid sequence, predict the 3D structure. So that has been a game changer in some areas. For example, if you want to design a small molecule drug that binds to a portion of a protein that you're trying to either activate or deactivate, this is now very much at the forefront of drug design. For our lab, we aren't designing small molecule drugs, but we were trying to design something called nanobodies. So some of you may have heard of nanobodies. So antibodies are a sort of complex 4-protein chain structure, and those antibodies bind to small epitopes or stretches of amino acids that are only about 6 or 7 amino acids long. And those antibodies, when they bind, for example, will neutralize a virus infectivity. What we were doing was using a version called nanobodies, which are simpler. They're just a single chain, and they're much, much smaller than typical antibodies. And because they're small, it means their amino acid, the areas of amino acids that vary among them, which change their binding properties, is a more limited repertoire. And so we were trying to use these prediction proteins, AlphaFold to start with, to predict nanobody sequences that would bind to an epitope of our choosing, or a sequence of our choosing. So that's how we were trying to use it. And it didn't work. And the reason is there's not enough training examples of nanobodies bound to their targets. You need a large training set of training molecules or structures to predict. And so that's how we tried to use it. And so far, there's just not enough training to give the program the ability to make predictions. So that's where we were at. The second thing I hope that we will do is use it for gene regulatory networks. Some of you, again, may be more familiar with transcription factors. They bind to specific sequences on the DNA to turn genes on or off. And we are looking at, again, the retinal cells and how they develop. And as part of those projects, we find transcription factors that are important for telling a cell to be, for example, a rod or a cone or something else. And those transcription factors that we identify as being crucial to that process are usually part of networks of transcription factors. So transcription factor A turns on B, turns on C. C may then turn on some genes that are important to make a rod. And so we've collected a lot of data of transcription factors and their target sequences, but we don't understand the network. And I know there's a lot of work in AI to address this problem, because anybody who works in development or even disease is interested in transcriptional regulation. So AI, I think, is going to make a big impact there. And we just haven't gotten far enough along yet in how to use it for that.
Thank you. Thank you so much. What was the name of that software that you said?
Old 2.
Okay, thank you.
There's more modern versions now too, but that was the foundational one from Google DeepMind. Anybody else?
Hi, I have a more general question for you. So, why were you initially interested in conducting research under the broader topics of neuroscience and genetics, like such as conducting research on retinal health?
Yeah, so as I mentioned, when I was transitioning to being a faculty member, I had previously been working in viruses. And the virus field was getting crowded. And I just didn't want to do work that everybody else was doing. You know, I wanted to feel like my contributions were not redundant. So I thought, well, what else can I do that's really interesting to me, where there aren't a lot of people trying to solve the same problem, but there's a lot of space to be innovative and make a contribution. And I thought neuroscience was that area, because it's probably still is one of the most, least understood areas. Also, I like developmental biology. And so I thought, okay, well, I could try to understand how all these neurons develop. And then I chose the retina because of some technical reasons, that we could take a needle with virus that, in our original case, we were using viruses to add genes to look at development. So when a mouse is born, the cells that we want to study are accessible. So it's a very easy inoculation. So we started with the eye because it was just accessible. But then I fell in love with it. I thought it was so cool. So we kept studying it.
Thank you.
Sure. I see more hands.
So I was wondering, since you said that certain genes are associated with eye diseases and other disorders like sickle cell, is it possible to have a genetic risk factor for a disease without actually having the disease? Like, for example, is there a gene that exists that only has like a 50% probability of causing a disease rather than, oh, there's a gene, there's most definitely going to be the corresponding
Yeah,
disease?
actually, most diseases in humans have that situation. So, the ones that are simple are the ones I talked about for eye disease. So if you have the disease gene, you will get the disease. Same for sickle cell. If you have the sickle cell mutation in one copy of your beta globin, you will get sickle cell. But the vast majority, that's not the case. So like I mentioned, AMD has what we call associated variants. So you might have a certain mutation, or I won't even call it a mutation. Let's just say a certain amino acid in a certain protein. It gives you a 5% greater chance of having AMD. That's it. And then let's say there's 10 such genes. If you're unlucky enough to have the disease associated mutation in 10 of them, then the probability adds up that you will get the disease. But most people don't have that bad of luck, right? It's just one. So most disease genes, or most, I should say, most human diseases are thought to be what we call multifactorial. And we just have an associated probability. So like if you go and do 23andMe or any of these, I don't know if any of you have done or heard of 23andMe, you can send your DNA into a company and they will tell you, oh, you have this particular amino acid in this particular protein. So you have a 1% greater chance of getting Alzheimer's at age 65. That's all they can tell you. So then you say, geez, should I worry about that? It's a 1% greater chance. Am I really going to get it? And the answer is probably 99% of the time, no. But you'll worry about that for the rest of your life. So I have chosen to not do that for that reason. But lots of people do. And you really, most of the time, from those sequencing companies, you're just going to get an associated probability due to your genetics of getting a disease, because that's most of the time the variants, the gene variants we know about are not what we call deterministic. It's not going to cause the disease 100% of the time. It's just going to give you a slightly greater probability of getting it somewhere down the line. That's most diseases.
Okay, thank you.
You're welcome.
So I remember you mentioned something concerning glucose shortage leading
Yeah.
to cone death or failure. So I was wondering if like diabetes could actually lead to that as well, or if like insulin production has any relation.
That's another great question. So yeah, it's a complicated area that we're trying to understand. So probably all of you know that when you have diabetes, you usually have more glucose in your blood. The other thing that can happen when you have diabetes is your blood vessels can get leaky. So there's actually retinitis pigmentosa and diabetes are common enough that there are people walking around who have both. Really bad luck. So in those people, they do have vision problems. And what happens is the blood vessels in the eye get leaky. And when they get leaky, it means that you're not getting good enough oxygen delivery to the eye and you're getting swelling. So inside your retina, you're now got all this fluid coming out of these leaky vessels and it's creating problems with your vision. The light can't get through properly. The cells don't get enough oxygen. And so you start to get what we call hypoxic, too little oxygen. Meanwhile, the rods are dying. And as we've talked about, more oxygen keeps coming in, even though the rods aren't there to consume it. So you have these two competing processes. In the end, you end up getting less diabetic retinopathy complications because when more oxygen comes in, it suppresses the growth of blood vessels. So you don't get as much new vessel growth and leakiness, which causes the swelling. So now the role of glucose there, we are not clear on. And I don't know if it would help cones survive to get more glucose. So many other bad things are going on with diabetes that we don't have an isolated way to look at it in people. In mice, what we did is we injected them with insulin, these retinitis pigmentosa mice, we injected them with insulin every day for 3 weeks, and the cones survived much longer. We're not still clear exactly how that worked, but it did work. Now, people are not going to inject themselves with extra insulin every day. Insulin has too broad of effect throughout the whole body. So it's not a good therapy. But if we understood what happens when we put insulin into the eye, we might come up with a therapy that's more directed at retinitis pigmentosa. So that's a hope.
All right, thank you so much.
You're welcome.
Professor, I had a question going back to sickle cell and malaria.
Yeah.
So if people with sickle cell are more susceptible to malaria, when they get gene editing and treatment to improve that, are they going to become suddenly more susceptible? Like, are they going to go from 50% to like suddenly 100%? And like, what precautions could they take? Because that would probably be like scarier.
Yeah. No, I think that's a great question. And yes, if they get enough correction to enough cells, they may become more susceptible to malaria or the downsides of malaria. And I'm not sure what the precautions are. I mean, there are other drugs that people take, and the number one defense for malaria that I understand is to not be exposed to the mosquitoes that carry it. So a lot of countries in Africa take pains to put nets over their beds at night and to try to avoid the really heavy malaria infested areas. And again, there are some medicines. So I don't really know beyond that what one does, but I suppose people who have been corrected, certainly in our country, we don't have much incidence of malaria. There is a little bit, but most people are not being exposed. So those folks are okay. And I don't think, as I said, we're going to be able to launch the sickle cell gene therapy in Africa at any high rate. I think we're going to rely on drugs for those cases. So maybe you stop taking the drug if you're getting, in a malaria infested area so that you still have the sickle cell effect.
Thank you.
Sure.
May I ask a question, Professor?
Please do.
Thank you so much for the amazing presentation. First of all, you are really an inspiration. I hope we can be like you. So my question was on, do you think, I know it's hard to replicate the human system, but if there were any simulations or AI systems that helped for example, sending your DNA and analyzing it, or for example, when you didn't need to experiment on mouse because these experiments continue for years, the simulation could just give you the results. There would be the computational cost, but would it maybe reduce the cost taken by the people who want to run this genetics test? So that, for example, for every pregnant woman, it would be mandatory and they would be treated like the babies, and there wouldn't be people not getting treated for diseases that can be cured.
Wow, what a great question. I mean, I don't know. You guys are going to be the future with AI and computational approaches. I think the places I can envision where this could be helpful is, I mean, some of the predictions are everybody will be sequenced. All your DNA is going to be sequenced. So people will know what you're susceptible to. And again, most of the changes in your DNA are going to lead to susceptibility, not direct predictions, but, probabilities of having this or that kind of disease. So that's going to happen. These gene networks that I mentioned, where there's a huge amount of complexity in how genes are regulated, how they turn on, turn off, what levels, and how they respond to environmental cues, those sorts of things. I think AI will make a big impact there. So predicting what's going to happen in a cell that has a certain genetic constitution might become real as AI gets more powerful. We still need to feed in a lot more data. As I mentioned, even with the nanobody structure, we just didn't have enough data to train the algorithms. So I think in terms of predicting how cells will behave when they have a certain genetic composition is still going to need a lot more primary data to feed in. But someday, I hope, basic scientists like myself will be able to provide more of that data, and then computational people will be able to use it to make predictions. I don't know how well, I guess, again, we need more data. If one could model, let's say, what happens in a cone cell when a rod dies, just for our case, how to blunt the effects or how to make the cell live longer. Maybe there will be predictions of what we should do and when, from understanding more what goes wrong inside the cone when their neighbors die. That might come out of AI, I hope. again, right now, we're just making guesses. You know, we see symptoms and we make a guess. We've actually tested about 100 genes to get to, I think, 7 now that really work well, but be great, as you said, if there's less time and money involved in testing mice, we could just say, okay, you know, here's a great prediction made by AI, let's test it. Ultimately, we have to test, I think, in animals, but to get fewer things to test, fewer animals to treat would be great. So hopefully you and the other person just asked me about AI. You guys got to get there. We got to give you more data, but then you'll have to work on it to give us good predictions.
Thank you so much, Professor. I wish I could be your student right now.
Yeah, I think that you'll find some really good labs to work in the future. Keep it up. Keep up your.
Thank you. Have a great day, Professor.
Yeah, you too.
Hi, can I ask a question?
Please.
Okay, so for my project idea, I had like the same train of thought of like I didn't want to do something that's been done a ton and a ton. So I'm trying to see if there's an association of elevated liver enzymes with diabetic macular edema. And I couldn't find anything online like pertaining to it. So I was wondering, like, do you research with diabetes? Like, if so, what do you think about it? And do you have any recommendations?
Wow, what a great question. And I love your attitude. Go into some novel space and figure out something new. I don't know anything about that actually. So you're gonna have to tell me. Sorry, I don't know.
Okay, it was worth a shot. Thank you.
Yeah,
Hi, can I ask a question?
please.
So my project relates to electromagnetic field therapy. So my question was, has electromagnetic therapy ever been used for gene editing, or has it ever been researched?
Not that I know of. How do you use it? What do you do with it?
My project is that it's relating to cell regeneration.
Ah, yeah, no, I don't know of any. The only thing I can even think of is the way that we deliver naked DNA. So we grow plasmids in bacteria and then isolate the naked DNA. We don't put it inside a viral particle. We inject that DNA just like we do viruses into what's called the subretinal space. And then we put electrodes across the eye and we shock the eye transiently. The animal's anesthetized, so it's just a millisecond shock. And supposedly, pores open in the plasma membrane and the DNA being charged is driven in by the electric field we put on. So that's a delivery mechanism that we use. It's not something you would do in a human, but we use it in our animals. And that's the only thing I know that uses electricity or to do anything. And that's for basic science questions.
Great, thank you so much.
You're welcome.
Hey, so I have a more general question that I think more people would be able to benefit from. And it is, what advice would you give to aspiring researchers looking to make an impact in either the field of genetics or any field in general?
I always say go with your interest and your passion. That if you're really passionate about something or really curious to know something or to solve something, go with that. If you can identify it, you can't always identify it. So if you can't, expose yourself to a lot of different kinds of science, the way people do different kinds of experiments. There's really different approaches to questions. You know, if you're a geneticist, you look for genes that cause a phenotype. If you're a biochemist, you tend to look at what protein binds to what other molecule. If you're a structural person, you look at the structures. So there's really different ways to think about the same problem. You may gravitate towards one of those approaches, or you may just have a question you really want to answer and you'll use multiple approaches. So if you don't have an identified question or approach, just expose yourself to a lot of different approaches and questions as you go through your training. And hopefully you'll find something you want to dedicate your life to.
All right, thank you so much.
Yes, you're welcome.
Super quick, guys. I just want to be like really mindful of time. 3 more minutes. What do you think, Dr. Cepko? Maybe 2 more questions? 1 more question?
Yep, go for it, somebody.
Can I ask a question? Oh, sorry, go ahead.
Yeah, you could go first if you want.
So since like the diseases you talked about in the eye are like genetic, if someone got like an eye transplant, would that benefit them in any way since the illness might be able to like resurface if it's basically in their DNA?
Yeah, so people are trying to do that. So there's 2 approaches. People are trying to grow photoreceptor cells in the lab and then inject the photoreceptors back in the eye. or RPE cells. They can both be grown in the lab. So that's an approach being taken. And people do talk about replacing the whole eye. I personally don't think that's going to work because the way the eye communicates with the brain is by these axons that leave the eye, and they go to very specific locations in a very specific way. And I just don't think that's going to happen with a whole eye graft. But these transplants of cells, like photoreceptor cells or RPE cells, might work.
Okay, thank you.
Yes, you're welcome. One more, anybody?
Yeah, so I had a quick question, Professor. So I was wondering, you mentioned how like oxidative stress is the cause of many like neurodegenerative disorders, and I know there's like several medications that could like help work as a therapeutic approach, but I also know there's like neuroprosthetics and there's also like stem cell therapy that could also work as like an approach to treat such a disease. I was just wondering like what your insight is about these two treatments.
Yeah, so stem cells are really an interesting modality. So what I just mentioned, these RPE cell grafts or photoreceptor grafts come from stem cells. So people can grow the stem cells in the lab, make the cell type they want, then put them back in. So again, that's a stem cell derived therapy. And people are also thinking about stem cell grafts, just injecting stem cells somewhere and hoping that the environment instructs the stem cells to produce a particular type of cell. So that also is being done. I forget, what was the other part of your question? That was a stem cell question. What was the other part?
I was also wondering like what your insight about neuroprosthetics are.
Oh yeah, prosthetics are interesting, and I don't know that much about them. There are some for the eye that are trying to give vision. They're so far not great. They give very slow and crude vision, but it's better than nothing. There's actually gene therapy of a different sort that I didn't talk about called optogenetic, where you give cells in the eye the ability to detect light. And these are cells that are either the last photoreceptors to boost their ability to do that, or other cells like these ganglion cells that send the axons to the brain. People are giving them a protein that allows them to act like photoreceptors, and that seems to be working. The prosthetics that are being used are little electrode arrays that are put inside the eye, where the electrode array can also detect light and then stimulate the ganglion cells to send signals to the brain. So again, that's not great, but it's better than nothing. So that's a prosthetic that's being worked. For other prosthetics in the brain, I'm just not that familiar. I know people are working on same idea of having an electrode array that'll respond to neural signals and send neural signals. Same process is going on in the eye. I don't know how well they're working.
All right, thank you so much.
You're welcome.
Awesome.
You all had great questions. So keep up your interest, as I've mentioned. I hope that some of you will be able to follow your dreams and continue to be interested in science and contribute.
Can we just say a huge thank you to Dr. Cepko for her time and insights today. I mean, this was honestly an amazing talk. I was taking notes throughout as well. Just really, really enjoyed it. So thank you so much, Dr. Cepko.
Absolutely, my pleasure. Bye-bye.
Thank you so much, Dr. Cepko. That was really informative.
Great.
Yeah, that was amazing.
Yeah, thank you a lot. I really appreciate it.
Thank you.
Yeah, thank you so much. Really appreciate it.
Thank you so
Thank you.
much.
It was very informative.
Thank you. Have a
Thank
nice
you.
one, everybody.
Thank you so much for everything, Professor.
Ariane, just saw your question. I'm gonna ask Dr. Cepko.
Okay, yeah, thank you.
Yeah, of course. There's some proprietary data in there, so it might take some time, or she might not be comfortable with sharing the whole thing, but I'll definitely ask her.
Okay, thank you.
Of course, of course, of course. All right, so yeah.